Hello dear students, myself Bhavani Pintela, working as an assistant professor in Noida Institute of Engineering and Technology Pharmacy Institute. In today's lecture, we are going to discuss about the transmembrane enzyme-linked receptor and JAK-STAT pathway. So, what is a transmembrane receptor? So, in previous lecture, before discussing about these transmembrane enzyme-linked receptor, let me see about the GPCR receptor, which we had explained in the previous session, right. I had explained about the ion channel as well. Ion channel, Z protein and enzyme linked receptors are the receptors which are present on the cell surface area. So, what is the difference between the Z protein and enzyme linked receptor? If I am talking about the Z protein, it consists an extracellular domain with for a ligand, and it consists of an intracellular domain for a Z protein. So, once the ligand bind on some other part of an extracellular, and there will be some changes happen inside the domain like G protein, and there will be conformational changes will be happen with that particular receptor. So, what is a Z protein receptor? When the Z protein receptor bind to an extracellular ligand, and it will activate the protein. Which protein it is going to activate? The ligand will going to activate the protein Z protein, okay, and this is called as an GPCR receptor. And what is an enzyme linked receptor? Enzyme linked receptors are bind to the extracellular. So, it also having the extracellular binding domain. Here the drug or ligand will bind to the extracellular ligand binding domain, and which will be linked to the intracellular domain, which consists of a catalytic activity. Okay, and if I am talking about the JAK-STAT pathway, so it is an enzyme activity which not having catalytic action. So, if I am talking about the tyrosine kinase activity, the tyrosine kinase receptor which having the catalytic activity. Okay, so it consists of an extracellular domain for a binding to that ligand, and which will be connected, which will be linked to the intracellular domain. Okay. And here I would to introduce the two terminologies like a protein kinase and phosphatases. So, what is a protein kinase and what is a phosphatases? Both will act in a opposite to maintain the homeostasis. If I am talking about the protein kinase, so what is the main function of protein kinase? The protein kinase, the function was to add up the phosphate molecule. It will phosphorylate the cell, it will add up the phosphorate, it will add up the phosphate group to the particular cell, and which is important. So, if I am talking about the antagonist drugs to the phosphokinase, the antagonist drugs are very important in a cancer therapy. So, these are very important class which are used in the cancer. And what about the phosphatases? Phosphatases, it will break the phosphate. So, phosphokinase will add up the phosphate, and phosphatases will lyse the phosphatases, and both will works in opposite in action to maintain the homeostasis. And here in this slide, I would like to differentiate the Z protein coupled receptor with that of an enzyme linked receptor. So, if you see here, GPCR receptor and enzyme linked receptor both are cell surface receptor. If I am talking about which type of receptor it is, both are cell surface receptor. So, in Z protein coupled receptor, the receptor present on the surface of a cell, and in the enzyme linked receptor also the receptor present on the surface of a cell. And second one, what is the energy molecule utilized by the GPCR? The energy molecule utilized by the GPCR, the energy molecule in GPCR was GTP, and in enzyme linked receptor the energy molecule was ATP. GTP stands for guanine triphosphate, and ATP stands for adenosine triphosphate. And what is the binding domain? If I am talking about the single from the single ligand binding, it triggers the cell activity. If I am talking about GPCR, when the ligand, if I am talking about the signal ligand binding, it triggers only one cell response. When the ligand bind to the particular Z protein coupled receptor, it will trigger only one type of a cell, but not in the enzyme. So, when the ligand bind to the enzyme linked receptor, it will trigger the multiple cell responses. Okay, ligand binding will activate. When the ligand bind to the Z protein coupled receptor, it will activate the Z proteins, and when the ligand bind to this enzyme linked receptor, it will activate the enzymes. Okay, and linked enzymes, you can see the linked enzymes in the GPCR? No, you cannot see the linked enzymes which are present in GPCR, but there are the linked enzymes which you can call as any kinase, which are present in the enzyme linked receptors. And what is the structure of any GPCR receptor? The structure of any GPCR receptor is a 7 transmembrane, and what about the enzyme linked receptor? It is a single alpha helical region. So, this is the differentiation between the G protein coupled receptor and similarities of any enzyme linked receptor. So, you can see some similarities and you can see some differentiation of any GPCR to that of any enzyme linked receptor. So, both are the cell surface receptor. In the energy molecule in G protein was GTP, in enzyme linked protein the energy molecule was ATP, and here when the ligand binds you can see the which will triggers only one cell response, but when you can see with the enzyme linked receptor it will triggers the multiple cell responses. When the ligand bind to the GPCR it will activate the G protein, and in enzyme linked receptor when the ligand bind to its enzyme linked receptor it will activate the enzymes. And what the enzymes linked, is there any linked enzymes in GPCR? There is no linked enzymes in GPCR, but there is a kinase enzyme linked to the enzyme linked receptor. So, it is if I am talking about the GPCR, it is a 7 transmembrane helical in structure, and if I am talking about the enzyme linked receptor, it is a single alpha helical region. It consists of a single alpha helical region. Okay, so, we will see the role of an enzyme linked receptor, how the enzyme linked receptors are important in our body. So, what is the main role of enzyme linked receptors? So, tyrosine kinase receptor is falls under in these receptors. Cytokines are also enzyme linked receptors. So, what is the main role of this enzyme kind receptor? It plays an important role in apoptosis, it plays an important role in cell differentiation, cell division, and plays an important role in cell growth. It plays important role in immune system responses, inflammation, tissue repairing. In all these conditions, the enzyme linked receptor will play a major role. And how many types of an enzyme linked receptor has been present? Depending upon the catalytic activity, the enzyme linked receptor having the 5 subfamilies. What are the 5 subfamilies according to the catalytic activity? So, we having the 5 subtypes of enzyme linked receptors are, first one is receptor tyrosine kinase. First receptor is a receptor tyrosine kinase, and second one is a tyrosine kinase associated receptor. First is a receptor tyrosine kinase, second one is a tyrosine receptor, tyrosine associated receptor, third one is a guanylate cyclase receptor, fourth one is a tyrosine phosphatase receptor, fifth one is a serine kinase receptor. We having totally 5 subdivisions or 5 subfamilies of an enzyme linked receptor depending upon its catalytic activity. So, enzyme linked receptors are sometimes you can call as a tyrosine kinase receptor or peptide receptors. Okay, the enzyme linked receptor sometimes you can call as a tyrosine kinase receptor, you can call as a peptide receptor. If you are moving about the structure of a tyrosine kinase receptor. So, what is the structure of tyrosine kinase receptor? Tyrosine kinase receptor consists of a domain. So, it consists of a ligand. First, what is the structure? First one, it consists of a ligand domain. So, what the ligand domain here? This is the receptor having the ligand. So, you can see here this is a receptor, this is a domain. So, this is a ligand binding domain, and it consists of a transmembrane alpha helix domain. Second one is transmembrane domain. It consists of a tyrosine residues. So, see here this one is a tyrosine residues, and this one is a tyrosine kinase domain. It consists of the structure of a tyrosine kinase receptor consists of a ligand binding domain, transmembrane domain, and third one is tyrosine residues, and fourth one is tyrosine kinase domain. So, this is a structure of a tyrosine kinase receptor. See here, this is a structure of a tyrosine kinase receptor. This is a ligand binding domain. This is the ligand binding domain of the receptor is connected to the transmembrane alpha helical domain, and which consists of an intrinsic intracellular domain like tyrosine kinase domain, and consists of an tyrosine residues. These are the structure of an enzyme linked receptor: ligand binding domain, transmembrane alpha helix domain, tyrosine kinase domain, and tyrosine residues. So, what are the steps involved in the enzyme linked receptors? So, what will be happen once the ligand bind to the receptor? What the steps are going to see once the ligand bind to its receptor? So, if you see here students, once the ligand, once the cytokinin or once the growth hormones, whatever it may be present to its receptor, then it will causes the dimerization. Dimerization of a receptor leads to the autophosphorylation. Once the enzyme has been get autophosphorylated, it will recure the 5H2 domain, and it will phosphorylate the 5H2 domain, and it will activate the transcription factors, and you can see the activation of transcription, right. So, this is a diagram where we can explain in a better way how the enzyme linked receptor, it will shows its action, how the cellular effects will be seen by the enzyme linked receptor. So, see here students, this is a growth factor. So, what it is? It is a growth factor. Growth factor is an example falls under enzyme linked receptor. If there is no growth factor bind to its receptor, if there is no ligand, you just imagine if there is no ligand to its receptor, the receptor will be present in a monomeric form. Now, the drug, now the ligand growth factor bind to the growth hormone. This is a growth hormone receptor. Which type of receptor? It is a growth hormone receptor. Now, the growth factor has been bind to the receptor, and what is going to happen once the growth factor bind to its receptor, the monomer and next monomer comes dimerization, and this dimerization, so see here in first step what is going to happen once the drug bind to its receptor, what is going to happen? If there is no drug to its receptor, the receptor will be stay in the monomeric form. Now, the drug bind to its receptor, now the receptor will causes the dimerization, conformational changes will be seen inside the receptor, and this one monomeric form will come and bind to the second monomeric form to its adjacent side, and now it causes the dimerization of a receptor. Once the dimerization, this is the second step, right? This is then a second step. Once the dimerization has been formed, when the drug bind to its receptor, the monomeric form combined with the next monomeric form, and it causes the dimerization. Once the dimerization has been seen, and this dimerization will activate the tyrosine receptor, and which will causes the autophosphorylation. Now, the receptor has been activated, after forming of a dimerization, the receptor has been activated, and what is the third step you can see here? The third step was autophosphorylation. So, if you see here students, so this is a tyrosine residue. See here, this is a tyrosine residue, this is a tyrosine residue. So, what we have to do here? We have to phosphorylate the tyrosine residues. So, without dimerization, you cannot phosphorylate the tyrosine residue. So, when the drug bind, the monomeric form and the next monomeric form will combine to form the dimerization, and once the receptor, after dimerization, the receptor will get activated, and this activated receptor will causes the autophosphorylation. Autophosphorylation, now the receptor will get phosphorylated, autophosphorylated. See here, this is a tyrosine residue. In the previous steps, you can see this is a tyrosine residue, this is a tyrosine residue. Now, the receptor has been activated, now the tyrosine receptor has been phosphorylated, which I had represent with the P. Now, the tyrosine residues has been phosphorylated. So, you can call it as an autophosphorylation. Now, the receptor has been autophosphorylated. So, tyrosine receptor has been autophosphorylated and the tyrosine residue has been phosphorylated. And what is going to happen here? And this is the third step, and after autophosphorylation, then it will phosphorylate the SH2, it will require the SH2 domain. What is an SH2? SH2 is any domain where if you see here GRB2 is in a protein, and GRB2 consists of an SH2 domain which consists of an tyrosine residue which have to get phosphorylated. Okay, and this is an important protein which will involve the further transcription process, and this GRB2 is a protein which is in inactive state which consists of an tyrosine residues, and now this autophosphorylated tyrosine receptor will attract the GRB2 protein, and now this attracted GRB2 protein will go and bind to the phosphorylated site of a receptor, and this phosphorylated site, when it go and bind to the phosphorylated site of a receptor, now the GRB2 which consists of an 5H2 domain will be get phosphorylated. Look at here you see here this GRB2 consists of an tyrosine domain, this is a tyrosine domain 5H2 domain, and which is which should be phosphorylated. And now when this phosphorylated when this GRB2 has been moved to the phosphorylated site of an receptor, now this GRB2 also get phosphorylated, and this phosphorylated GRB2 will activate the RAS pathway. And how it will activate the RAS pathway? This RAS pathway will be present in an inactive state. Normally the RAS will be attached with the GDP. Once GRB2 has been get activated, this GDP will be replaced with the GTP. Now the RAS has been get activated, and this activated RAS will be phosphorylate the RAF, and the RAF will in turn phosphorylate the MEK, and MEK will in turn phosphorylate the MAP kinase, and finally you can see the transcription factors. And what will be happen when this GRB2 protein has been activated, which in turn activate the RAS, RAF, MAC and MAF pathway, and finally you can see the gene transcription. So, what you can see here? You can see the gene transcription by the enzyme-linked receptor. So, what is the enzyme-linked receptor? I will explain you here one more time. When the drug bind here, what is the drug here? Growth factor. Growth factor is a drug bind to its receptor, then the receptor of monomeric form come and bind to the second monomeric form, and this monomeric form of both receptors will causes the dimerization. Okay. If imagine students, if there is no drug bind to its receptor, the receptor will be stay in the monomeric form, no doubt in it. And once the drug has been occupied to its receptor, the monomeric and the next monomeric will form the dimerization. When is the dimerization? This is the second step. What is the first step? Ligand binding. What is the second step? Dimerization. What is the third step? Autophosphorylation. What is the fourth step? Requirement of 5H2 domain. And what is the fifth step? The activation of an 5H2 domain. When this GRB2 domain has been phosphorylated, in turn it will activate the cellular changes. So, see here, when the drug bind to its receptor, you can see the dimerization of a receptor. Once the receptor has been dimerized, the receptor has been activated. Once the receptor has been activated, the receptor consists of tyrosine residues. Now, once the receptor has been activated, the tyrosine residues will get phosphorylated, which you can call as an autophosphorylation. Why? Because the own receptor, the self receptor consists of an tyrosine residues. Here the self tyrosine residue has been get phosphorylated. Once the receptor get activated, the next step was autophosphorylation, and this tyrosine residues has been get phosphorylated, and this phosphorylated tyrosine molecule or tyrosine residues has been phosphorylated, and this tyrosine kinase receptor will attract the GRB2 protein, because the GRB2 consists of 5H2 domain, which consists of an tyrosine domain. What is GRB2? GRB2 is a protein which is present inside the body. The GRB2 is a protein which is an inactive form. Once the molecule, once the receptor has get activated, once the receptor has get autophosphorylated, this GRB2 consists of SH2 domain, which you can call as an tyrosine residues. The GRB2 consists of a tyrosine residue, and this tyrosine residue of GRB2 will be get attracted to the phosphorylated site of a tyrosine kinase receptor, and now this GRB2 will go and migrate. This GRB2 will migrate to the phosphorylated site of a tyrosine kinase receptor, and once GRB2 will go and bind to the phosphorylated site of a tyrosine kinase receptor, what is going to happen here? Here the GRB2 protein, which consists of an tyrosine residues, has been get phosphorylated. Now this protein, now this tyrosine residue also will get phosphorylated, and this phosphorylated GRB2, which in turn phosphorylate the RAS, RAF, MAC, MAP, and finally you can see the cellular effects. Okay, next we will discuss about the JAK-STAT pathway. So, just what is the difference between the enzyme-linked receptor like tyrosine kinase receptor, and what is the difference between the JAK-STAT pathway? So, one type of receptor which consists of an intrinsic activity, both are the enzyme-linked receptor, no doubt in it. JAK-STAT is an enzyme-linked receptor, tyrosine kinase is also an enzyme-linked receptor. One type of receptor consists the catalytic action with the enzymatic action, the enzyme which consists catalytic action, and the JAK-STAT do not consist of a catalytic action. So, if I am talking about this JAK-STAT pathway, what is the full name of JAK? JAK stands for JA, Janus, K stands for kinase. Okay? And what is mean by STAT? STAT. signal transducer and activator of transcription. So, STAT stands for, what is a STAT? It is a signal transducer. It is a signal transducer and activator of a transcription. It will activate the transcription. So, JAK is a kinase, STAT is it is a protein which will activate the transcription. So, this is the reason why you can call it is a JAK-STAT pathway. So, these are the pathway which are the major pathway for transferring the signal from the cell membrane receptor and to the nucleus, and which will place an important role in the immune system. So, in this pathway the cytokinins go and bind to its receptor and it will shows the cellular changes inside the cell. And if we suppresses the cytokinin receptor, there will be suppression of an JAK-STAT pathway. There will be no formation, there will be no phosphorylation of an STAT. So, here the STAT is a protein. Here the STAT have to get phosphorylated. Once the STAT has been phosphorylated, then it will be get dimerized, and this dimerized STAT will causes the intracellular changes. Okay. So, the JAK-STAT is a pathway which will in turn causes the transcription pathway. So, what is a JAK-STAT binding receptor? So, if you see here students, the JAK-STAT activated upon when the JAK will get activated, the JAK is an inside it is an kinase, it is an intracellular kinase. JAK is an intracellular kinase which will be get activated when the ligand bind to its receptor. So, what is the ligand here? The cytokinin is a ligand. When the cytokinin stimulate its receptor, the JAK will get activated. Once the JAK get activated, the JAK will phosphorylate the STAT, and this STAT will get dimerization, and this STAT will translocate inside the nucleus and causes the cellular changes. Okay, if I am talking about the JAK with having the families in mammalians, the JAK having the 4 members like JAK1, JAK2, JAK3, JAK4, and you can call tyrosine kinase 2 TYK2. And what about the STAT? The STAT family has been composed of 7 members. So, what are the 7 members of an STAT? STAT1, STAT2, STAT3, STAT4, STAT5A, STAT5B, and finally STAT6. So, here if I am talking about the JAK, it consists of 5 members, it consists of 4 members, and in STAT it consists of 7 members. The JAK members are JAK1, JAK2, JAK3 and tyrosine kinase 2. And what about the STAT? STAT members are STAT1, STAT2, STAT3, STAT4, and the STAT5 has been divided into STAT5A and 5B, and finally STAT6. These are the 7 members of an STAT members. So, here in this slide we are going to see what is how the JAK-STAT will get activated, and if there is any lack of JAK-STAT pathway, what is going to happen, and what are the examples falls under the JAK-STAT. First we will see the what are the examples. First we see the what are the examples of JAK-STAT pathway. So, gamma interferon, alpha interferons, erythropoietin, growth hormone, prolactin, interleukins. So, all these are the examples which falls under JAK-STAT kinase. So, if I am talking about the JAK-STAT pathway, all these are the examples. What are the examples of JAK-STAT? Alpha interferon, gamma interferon, erythropoietin, growth hormone, prolactin, interleukins. So, what is happening to here? So, if you see if the JAK-STAT pathway get activated, you can see some changes. If there is lack of a JAK-STAT pathway, what is going to inflammation, and it will causes the gigantism. So, all these are the effect with the lack of JAK-STAT pathway. So, if you see here, agonist drug, when the agonist drug bind to its receptor, it will increases the affinity of a tyrosine protein, which you can named as a JAK-Janus kinase, and this JAK-Janus kinase will be get activated, and it will get phosphorylated, and it will binds to the STAT, and this STAT will get phosphorylated, and this STAT phosphorylated, it causes the dimerization, and it will be interlocate into the nucleus, and will causes the some cellular changes. So, look at diagram here, I will explain you in detail. So, this is a cytokine. When it bind to its receptor, this is a protein, JAK protein. It is a tyrosine molecule which is present inside, and JAK consists of a tyrosine residue. Once this monomer has been get dimerized, the JAK will get phosphorylated. The monomer come together to form the dimerization, same like enzyme-linked receptor, tyrosine kinase receptor. Now dimerization has been seen, and once the dimerization has been seen, what is going to happen here? The JAK consists of a tyrosine residues will be get activated and it will get phosphorylated. Once it get phosphorylated, what will be happen? Now the STAT, it is a protein will attract to the JAK pathway, and which consists of a tyrosine residue, and this tyrosine residue of a STAT will go and bind to the JAK protein with JAK protein of an phosphorylated site, and now this STAT will get phosphorylated, and this phosphorylated STAT will forms the dimerization, and this dimerization of an STAT will interlocate into the nucleus and will causes the cellular changes. Thank you.